Hello my dear students, today we will start the first chapter, that is heredity and evolution, from your science 2. Now we all know that transfer of biological character from one generation to another generation via gene is known as heredity. You have studied this in 9th standard. Few scientists I would like to name here because we have to know that how it all started. So the first scientist that you all know, that Johann Gregor Mendel, he was the pioneer of modern genetics, and you all have studied that he has done experiments on pea plant, and he found that how the genes transfer from the parent pea plant to the offspring, right? Now there were some changes also. That changes happens because of the mutation and other factors that was put forth by the theory of Hugo de Veris. You will study it in higher classes. Now the next scientist was Walter and Soton. They were two scientists. There were two scientists who first found that chromosomes are found in the pair, and in grasshopper they first did this experiment and found out this, so one coming from the male and another coming from the female for specific character. Now all the living beings except viruses, DNA is the main genetic or hereditary material that was found by the three trio scientists. They are Oswald Ivory, Macleay McCarthy and Colin McLeod. And in 19 who first discussed and who first gave us the information that proteins are synthesized inside the bacterial cell. It is not just that the protein we take inside and in the form of food is only the protein that we have inside. Also inside our body, also our cell membrane, many blood components, enzymes, hormones are composed of proteins. So our body needs to make those protein, those essential proteins and amino acid. For that we are going to study the protein synthesis in our body, how it happens and all that. Now the science of heredity is very useful for diagnosis, treatment and prevention of heredity disorder. Also the production of hybrid variety. There two different varieties are hybridized, and a new plant or new organisms are grown, animals or plants maybe, which will have both the qualities, both the good qualities in them. So we will see further this in this 10th standard. You will study more about it. And also in some food factories and vaccine and hormone production companies are there, where this industrial use of microbes are very much famous, right? So because of this we all have to understand all this heredity and DNA and why protein are synthesized, what is the basis, and what information are present in the DNA. All this we will study here. Now coming to the protein synthesis, I will take you to 9th standard, where you have studied the structure of DNA, which is double helical structure, and there were nucleotides present: A, T, G, C. A stand for adenine, T stand for thymine, G stand for guanine, and C stands for cytosine. Now A forms the bond with T, that is adenine, thymine and guanine and cytosine. Now you also have studied RNA, the three types of RNA, and they are also nucleotides. These nucleotides are present only with the exception that in place of thymine, uracil is present. So A doesn't form the bond doesn't form with T because thymine is not present in RNA, but uracil is present. So adenine forms the pair with uracil. So remember all these concepts because those are important points to be remembered. Now the protein synthesis starts in the nucleus, okay, because the DNA is present inside the nucleus. Now the first thing that happens is the uncoiling of the DNA. Uncoiling meaning DNA is present in double helical structure, so just one strand of the DNA take part in protein synthesis. So for that, the first enzyme that comes into the picture is RNA polymerase, that binds with the DNA, and hence it unfolds or uncoil the DNA and making one strand as a template. Now in that strand many nucleotides will be present. So based on that nucleotides, the new nucleotides will be brought and a chain of mRNA will be made. Okay, so for A, U will be made, for G, C will be made, and henceforth. So the long DNA strand is acting as a template, acting as a platform on which the new nucleotides comes and they get synthesized. So it is like a chain of so like that it goes on, and after completing this mRNA, which is newly made, which is complementary to the DNA which comes out from the nucleus, and the protein synthesis, the actual protein synthesis process starts in the cytoplasm, which is outside the nucleus. Now the process through which the DNA acting as a template and mRNA is being made, that process is known as transcription. Okay, so making of mRNA through the DNA is known as transcription. Now this long chain of mRNA has thousands of triplet of codon. Now why am I calling it as codon? Because the nucleotides now will be, I'll be calling as codon. 3 nucleotide act as one codon. Okay, now they will bind with the rRNA, that is ribosomal RNA, and that will initiate the tRNA to bring the amino acid as per the nucleotide present on the mRNA. So the amino acid is also coming in the triplet form. Okay, triplet, why I'm calling 3 nucleotide equal to one triplet of codon. Like if they have U, A, G, so the anticodon that will be brought by the tRNA, for U it will bring A, for T it will for A it will bring U, and for G it will bring C. So codon is present on mRNA and anticodon is brought by the tRNA. Okay, and this process of bringing and attaching the nucleotides, codon or anticodon rather you say, is known as translation. The process of bringing of amino acid by the tRNA and forming a bond. Which bond is formed, student? Here peptide bonds are formed. The nucleotide between the tRNA which is brought and the nucleotide present on the mRNA, the bond formation between these two nucleotides is peptide bond. Okay, and this process is known as translation. Okay, now this rRNA shifts by the one codon, meaning one codon the things gets over, the tRNA has brought the amino acid, now the rRNA will shift towards in forward direction. The shifting of rRNA so that the process gets continues and the tRNA brings the amino, keeps on bringing that amino acid, that has to happen, right, till the entire mRNA is being coded, the amino acid is brought about by the tRNA and the process gets complete. So this shifting of RNA is known as translocation. Now three words I have told you here: transcription, translation and translocation. Transcription is nothing but formation of mRNA through the DNA. Okay, now translation is nothing but the bringing of amino acid and attaching it with the mRNA by the peptide bond, that is known as translation, and shifting of rRNA through the one codon is known as translocation. And now like this entire mRNA chain is being coded and the amino acids are being formed. Like thousands of amino acids are formed, and this complex protein is now manufactured, which can be used by the cell. Now there are 20 amino acids are being coded by the Indian origin scientist Dr. Harvind Khurana, and he was awarded with the Nobel Prize in 1968. Now during this all process there could be some mutational change. Mutational change meaning in place of some nucleotide, some different nucleotide came and formed the peptide bond. So these changes are very minor and it is not like of great concern, but there could be some major mutation happening where one amino acid is completely changed by another amino acid, for example in sickle cell anemia, if you remember. So because of that, this complete the chain of amino acid get changed and it behaves in a certain way. Okay, so that is considerable, that could be fatal for any organism which receives such kind, or any cell which receives some such kind of mutation. So till here only I'll keep, students. I would link some video for you so that you can understand this whole process of protein synthesis. Okay, any doubts, anything is there, then you can send me the feedback.